Okay, so welcome back to this next video in which we are discussing heterotrimeric G proteins. Okay, so we're currently in the process of discussing the prenylation of the G gamma subunits. Okay, so we've discussed that for a protein to be prenylated, what it needs to have is a CAX box. It needs to be a CAX box protein, which is just any protein with a CAX box. Okay. And to have a CAX box, what you need to have is in the final 4 amino acids within the polypeptide, you need to have the 4th amino acid from the final amino acid as a cysteine. The 3rd and 2nd from last amino acids need to be aliphatic amino acids such as glycine, alanine, valine, leucine, or isoleucine. And then the final amino acid in the polypeptide determines which of the 2 prenyl groups you're going to add on, whether it will be a farnesyl group or a geranyl group. So, it's now time to discuss what the structure of these prenyl groups are. So basically, prenyl groups are based on the structure of isoprene. So I'm going to show you the structure of an isoprene molecule, and then we'll discuss how you can make polymers of isoprene, and how the farnesyl and geranyl groups are just polymers of isoprene. Well, they're oligomers really, they're not quite polymers. They'd need to be longer to be considered polymers. Okay, so let's start with the structure then of isoprene. So we'll start with the molecular structure and then we'll draw out a skeletal structure. Okay, so isoprene has a main backbone of 4 carbons. Okay, and you have a double bond here and a double bond here. And then off this carbon you then have a methyl group. Okay, and then we can saturate everything else with just hydrogens coming off here. So 2 off here, 1 off here, and 2 coming off up here. Okay, so this is the molecular structure of isoprene. Now if we want to draw the skeletal structure, then it's a lot simpler. Okay, so in skeletal structures we don't show carbon atoms, they are implicitly shown as corners, and we don't show hydrogen atoms coming off carbon atoms, so you can imagine that this is therefore going to be a very simple skeletal structure. Okay, so here are the 4 carbons that make up the backbone, okay, and then you'll have a double bond here, a double bond here, and then you'll have a methyl group coming off like so. Okay, so I'm just going to invert this and put it the other way up for now. Okay, so I'll draw it like this as well. This is the way up that we're going to use it when drawing out the farnesyl and geranyl groups, although obviously it's trivial. Okay, so how can you polymerize isoprene molecules together? Well, basically, if you imagine breaking the second of the 2 bonds in the double bonds between these 2 carbons and these 2 carbons, like so. Okay, so break one of the 2 bonds, not both of them, only one of them. Okay, now one covalent bond contains 2 electrons, 1 from each of the 2 members of the bond. Now, this isn't an electronic mechanism, but imagine returning 1 electron to this carbon and 1 electron to this carbon, and in the case of this bond here that we're breaking, let's return 1 electron to this carbon and 1 to this carbon. That means that all 4 of these carbons have a single free electron. Then you combine this carbon to this carbon, because they both have free electrons, to create a double bond in the middle here, and that will leave both of the end carbons with free electrons. So our imaginary intermediate, if you like, will be as follows. Okay, so here's the 4 carbon backbone, here's the methyl group coming off the edge, you've got a double bond there, and now these 2 carbons on the end both have free electrons. So you have a free electron there and a free electron there, okay? So then what you can do is you can take multiple of these imaginary intermediates and bind them together. So if I take another one here, like so, then this carbon here will have a free electron as well. It combined it with this carbon's free electron to make a bond there. Now if we connect 3 isoprene molecules together, this is what a farnesyl group is. Okay, so let me do that. So let's put 3 of these intermediates, if you like, next to each other, and then we'll join them together. So here are these intermediates where each of the end carbons has a free electron. Okay, and here's our final one here. And now what you can do is you can link this carbon to this carbon and this carbon to this carbon. And you might ask, well, what are you going to do about the two end carbons? Well, this one is what's going to bind onto our protein, okay? So this will be a bond to something arbitrary that we don't know yet. And we're going to stick a hydrogen off this carbon to make up that final electrons bond, basically. Okay, so that then is what's known as a farnesyl group. Okay, and it's called a prenyl group because it's based on the structure of isoprene. So any group that has been made out of isoprene molecules is called a prenyl group. Okay, so farnesyl groups are specifically prenyl groups where you've stuck 3 isoprene molecules together, basically. Now, geranyl groups are basically where you've stuck 4 isoprene molecules together. So let's do this now. So let's show the 4 isoprene intermediates, and then we'll bind them together. So here's one. Then we've got the second one here. Okay, the third one here, like so, and then the fourth one we'll put here. Okay, and then what we'll do is we'll bind this one to this one, this one to this one, this one to this one, and then we'll put hydrogen off the end and bind this one onto our protein, and this will be a geranyl group. So these are not molecules, they are groups, because they are going to be added onto something else to make a molecule. They're not complete of their own, on their own, basically. You need to bind them to something. Okay, so we have these farnesyl groups and these geranyl groups, and they're both examples of prenyl groups because they're both these oligomers of isoprene. You wouldn't quite call them polymers because they're only 3 and 4 isoprene molecules put together. An oligomer is kind of like an intermediate. It means a small polymer. So these will be described as isoprene oligomers. Right, okay, so you are now going to add farnesyl groups and geranyl groups onto proteins which have got this CAX box. So if we just put this in here, here's our protein which ends with a CAX box, like so. And depending on which amino acid you have at position X will determine whether you get a farnesyl group added or a geranyl group added. Okay, but where are these groups actually going to be added onto? Well, they're added onto the cysteine of the CAX box. So let me show this happening. So we'll draw that cysteine amino acid residue within the CAX box. Here's the amino group, here's an alpha carbon, here's the carboxylic acid group, and it will be linked to the amino group of the next amino acid along, which will be this A amino acid here. Okay, and then the R group of cysteine is a methylene group, and then with a phyl group coming off. Now what you're going to do to add either a farnesyl group or a geranyl group on is that you're going to split the bond between the sulfur atom and the hydrogen atom. Okay, imagine giving 1 electron back to the sulfur and 1 back to the hydrogen. And then what you're going to do is you're going to bind, let's say we're going to add a farnesyl group, but you're going to bind this carbon here, which remember was the final carbon of the farnesyl group, which still had a free electron. You'll bind that to this sulfur atom's free electron. Okay, and then you can imagine that the hydrogen is the hydrogen which then will go on to the other side, where I said we would need to put a hydrogen to saturate that final carbon on the other end. Okay, so what you're therefore going to end up with looks like this. Okay, so here's our cysteine residue again, and I'm now going to attempt to draw the farnesyl group on, but I haven't left much space for it. Okay, so here's the methylene group, which is going to have to squish in a bit, and I'm certainly going to have to draw a skeletal structure rather than a molecular formula. Okay, so we're going to have to squash it in this time. Okay, so there's our first isoprene group with the double bond there. Here's our second isoprene group here, double bond here, and then, yeah, it's easily fitted in. Here's our third isoprene group. Okay, and we'll join them all together to make a farnesyl group here. And now, this sort of a bond where you have a carbon atom linked to a sulfur atom linked to another carbon atom, that's called a thioether. And again, this draws parallels with the nomenclature for oxygen. So when you have an oxygen atom between 2 carbon atoms like this is known as an ether link. Now, I've already told you that oxygen and sulfur are in the same group periodic table and therefore have very similar chemical properties. So when you have a sulfur atom between two carbon atoms like this, it's called a thioether link. Okay, and you would now say that the cysteine has been farnesylated. Okay, or you would say that the gamma subunit of the G protein has been farnesylated, rather. Okay, now the same is true for geranyl groups. All that's different is that you have an extra isoprene molecule on the end, basically. Okay, so that then is the prenylation of the G-gamma subunits, but that's not all that happens to the CAX box. The story doesn't end there. Once you've added the prenyl group onto the cysteine of the CAX box, what follows is what's known as post-prenylation modifications. Okay, so the first post-prenylation modification that occurs is AAX proteolysis. Okay, so AX proteolysis. Okay, so remember that our CAX box protein, or our G-gamma subunit, had this CAX box structure at its carboxylic acid terminus. Okay, so here is this CAX box structure. We have now just added on a prenyl group onto the cysteine residue. Once you have done that, what you're going to actually do is cut off the final 3 amino acids. You're going to cut off the AAX, basically. You're going to split the bond between the cysteine amino acid residue and the third from last amino acid residue, which is this aliphatic amino acid. And this AAX little peptide fragment here is going to get cut off the rest of the polypeptide, okay? Which will mean that this cysteine amino acid, which has the prenyl group dangling off it, will become the final amino acid in the protein. Okay, that's the first modification you make. The second post-prenylation modification is that this final cysteine residue is then methylated. So methylation is going to occur. So, let me get another piece of paper and show what will happen here. Okay? Right, so, if I draw my protein now, so here's our protein. Here's the amino terminus of this G-gamma subunit, and now at the final terminus, what we currently have is this cysteine amino acid, and I will show the whole cysteine amino acid. So here is the amino group linked to the carboxylic acid group of the next amino acid along. Here is the R group, which has a methylene group, then with a thiol group, and then linked onto this is the prenyl group, which I won't draw, I'll just draw as a line there, okay? So you have either a farnesyl group or a geranyl group linked off there, okay? And now it's the final amino acid, so its carboxylic acid group is free, like so. Okay, now we've just done AAX proteolysis, which is why this cysteine amino acid is now the final amino acid. The next modification is going to be to modify this carboxylic acid group. So you're going to turn it from being a pure carboxylic acid group to being a methyl ester. So basically what you're going to do is take that hydrogen off that oxygen. So we're going to get rid of that, and we're going to replace it with a methyl group. So we're going to have this oxygen linked to a methyl group, like so. Okay, and that's what methylation means. You're going to turn that final carboxylic acid group from being a pure carboxylic acid group to being a methyl ester. Okay, so that's the lipid modification that is occurring to these G-gamma subunits of heterotrimeric G proteins. So what we've seen now is that all alpha subunits of heterotrimeric G proteins get at least one acyl group added onto them. Some even get 2, okay? And these acyl groups will anchor these alpha subunits in the inner leaflet of the phospholipid bilayer. Similarly, the gamma subunits also get a lipid modification. So let's just draw this out again. So we'll draw the same picture that we drew when we first started our discussion. So here is the phospholipid bilayer, the outer leaflet and the inner leaflet. And then sitting underneath it we have the alpha subunit of our heterotrimeric G protein here, okay, which is currently, let's say, in the off state and therefore has guanosine diphosphate bound to it. Okay, and it has either one or maybe two lipid molecules near its amino terminus, which will anchor into the inner leaflet of the phospholipid bilayer and maintain that alpha subunit of the heterotrimeric G protein at the inner leaflet of the phospholipid bilayer, so that it isn't just sort of in the cytoplasm of the cell, it's at a very specific location, which is just underneath the phospholipid bilayer. Okay, then you have this complex of beta and gamma subunits, which will be bound to the alpha subunit. So here's a beta subunit, and here's our gamma subunit, and the beta subunit doesn't have a lipid modification, so it relies upon the gamma subunit having its prenyl group. So the gamma subunit has this prenyl group here, so let's have that in orange. So the prenyl group can be shown in orange, and either the palmitoyl or the myristoyl group can be shown in pink over here. So the beta subunit must bind to the gamma subunit, which then has a prenyl group which anchors it in the membrane if the beta subunit is to stay at the inner leaflet of the phospholipid bilayer. And indeed, the beta and gamma never break apart, basically. You have to heat them up to non-physiological temperatures to make them break apart. Okay, so here's the beta subunit, here's the gamma subunit in blue here. Okay, and then we'll colour in the alpha subunit in red. Okay, so we've now discussed all the different types of alpha subunits, the different types of beta and gamma subunits. We've discussed this lipid modification, which is incredibly important. Do not dismiss this as trivial. That's how these are remaining in the position that they need to be in order to interact with G protein coupled receptors. In the next video, what we'll talk about is the G protein cycle. So what happens when a G protein coupled receptor becomes active, how it interacts with these, and then how they deactivate, basically.